Our next presentation is a double act. It's my pleasure to welcome Professor Anna William and Sunniva Bøstrand from the University of Edinburgh.
Thank you very much indeed. I'm Anna Williams, and I'm going to start, and hopefully my slides will turn up. Excellent. So we're going to talk to you today about glial-specific transcriptomic changes in Huntington's disease. And we thought we would tell you a little bit about ourselves because we're not the sort of people you tend to see at your conference. We've never been to a Huntington's disease conference before. And so we thought we should tell you about us. So we come from Edinburgh, and it's always this sunny in Edinburgh. But it is a very beautiful city. We work in the Institute of Regeneration and Repair, which is on the south side of the city. And we are mostly in the Centre for Regenerative Medicine that you see on the left. And that's closely aligned to the centre next door, which is the Tissue Repair Centre. And my lab has been there for about 10 years, and what we're very interested in doing is understanding and manipulating myelin pathology. So what we want to do is to try and maintain or restore oligodendrocyte health and myelin integrity, because we're interested in doing that to preserve neuronal and axonal function. And of course, we don't mind which neurodegenerative disease we do this in. We started the lab thinking mostly about multiple sclerosis. We're now thinking more about vascular dementias. And now we're thinking about Huntington's disease. But we are experts in oligodendrocytes. And I would like to point out that oligodendrocytes were 100 years old last year in 2021, from their first description, clearly. And for that time, we had a 100th birthday party for the oligodendrocyte when we had to wear hats with oligodendrocyte themes, and this is the group with their hats. So we're very focused on oligodendrocytes. So you're saying, well, why are we talking about Huntington's disease? Now, we know that in Huntington's disease, there's this exquisite vulnerability of the striatal mediospinal neurons, and that, of course, gives you the movement disorder. But as Mahmoud has been telling you, and lots of other people have been telling you over the last couple of days, there are prominent white matter deficits in HD, and this correlates with the symptoms. And you've seen this picture already. This is a picture showing in black myelin and showing the lack of integrity of myelin in HD in people. And why we're interested in oligodendroglia, or in fact glia generally in neurodegenerative disease, is because there's a therapeutic potential with these. We know that we can regenerate glia, they regenerate themselves, and so can we use them to help with therapies for neurodegenerative diseases, including HD. So I'm not going to dwell on this slide because Mahmoud has been telling you about this in much more detail, but I'll just summarize it for you, is that we know that if we manipulate oligodendroglia or astrocytes, or glia progenitors that can produce both of those in at least HD mice, we can change the phenotype. So these three pieces of evidence here, what's happened is that each group has downregulated the mutant Huntington in either oligodendroglia, the progenitors, or astrocytes, and found an improvement in the pathology, but also an improvement in the behavior. So we think that manipulation of glia might be useful in HD, at least in mice. But what about glia in humans with HD? So to look at that, we formed a collaboration with Josef Prilla, but also with the Netherlands Brain Bank and Leiden teams, and with Sunniva Bøstrand, who I'm going to introduce in a minute. And we decided to look at characterizing the changes to the transcriptome in glia in human HD. Sunniva is a final year PhD student, and I'm going to hand to her next.
So in this project, I've been working to characterize the changes to transcriptomic heterogeneity of glia in Huntington's disease using single nuclear transcriptomics. And I wanted to start off by introducing single nuclear transcriptomics in case not everyone is familiar with that. So many of you will be very familiar with this graphic showing the central dogma of biology, which tells us that DNA makes RNA and RNA makes protein. And as I've highlighted here, when we work with transcriptomics, we are looking at RNA. And we do that because at the level of RNA, gene expression is highly dynamic. So firstly, it's cell type specific in that different types of cells express different types of RNAs. But beyond that as well, the RNAs expressed in a cell can tell us something about their functions or the state that that cell is in. And so we use the technology that allows us to capture a single nucleus and to read out the information contained within its RNA. And we can do this at scale, so it's a high throughput method that allows us to look at hundreds of thousands of individual nuclei and read out this information, which tells us not only what type of cell that nucleus came from, but it can also tell us something about its function or its state. So it allows us to look at the cellular heterogeneity in the brain. And this is a method that has been used previously by us and other groups to look at changes to cellular heterogeneity in the context of neurological disease and in aging. And all of these studies that are listed here have shown that there is a change in the heterogeneity of glia in all of these neurological conditions, even those where a glial component hasn't really been recognized as part of the pathology previously. And so we know that this is a highly sensitive method that is appropriate to studying glial change in disease, and we wanted to apply this in the context of HD. And so in order to do that, we had to source post-mortem brain tissue, which we acquired from 3 different brain banks, the Netherlands Brain Bank, the Leiden University, and also Edinburgh Brain Bank. And we sourced brain tissue from HD patients and age and sex matched controls from these 4 regions here. We looked at the caudate nucleus, the frontal cortex, the hippocampus, and the cerebellum. And we acquired this tissue from 6 HD donors, and then we paired that with age and sex matched control tissue. And this here is just to show you the number of CAG repeats of these HD donors, where you can see that they mostly cluster around an average in the low 40s, whereas there's 1 donor with a longer CAG repeat length of 54. And so the key questions that we were trying to answer with this project is firstly, whether glia are different in HD compared to controls. And as the speakers in this session so far have introduced you to, we have very good reason to believe that that is the case. And we also wanted to look more deeply into that to see whether there are glial signatures that are more or less common in HD. And as I mentioned, we included 4 different brain regions because we wanted to address whether these changes can be seen globally and if there are regional differences. And then finally, we also wanted to probe whether there might be future targets for manipulating glial function in HD. And so very briefly, the way that we carried out this experiment is that we started off with the fresh frozen post-mortem tissue, and we extract single nuclei from them, and then we barcode these using the 10x genomics platform, and we sequence the RNA, and then we process this through a fairly standard pipeline of data processing and analysis. And then at the bottom here, you can see some of the most common and useful types of analysis that we can do with this data. So we can firstly cluster the individual nuclei together based on their similarities in their gene expression. And then we can also look at differential abundance, which tells us something about whether certain populations are more enriched or depleted in HD. And then similarly, we can look at differential expression, which tells us whether there are certain genes that are up or down regulated in disease. And this here is a representation, a UMAP representation of the data that we arrive at the end of that pipeline. This includes all of the brain regions and all of the donors, just with the clusters grouped in by their broad cell type. And then here on the left is showing you the clusters in the caudate that contain cells expressing PPP1R1B, identifying them as medium spiny neurons, and we see a drastic reduction in these as we would expect. And then because we're interested in the glia, we extract from this data the astrocytes, the microglia, and the oligodendroglia, which consists of oligodendrocytes, oligodendrocyte precursors, and also committed oligodendrocyte precursors, or COPs. And then we subcluster each of these glial types in order to group them into smaller groups based on their similarities in their gene expression, so that we can look at a higher resolution and detect more subtle changes in these. And just a note, we have annotated all of our glia in a very specific way because we wanted to avoid any assumption of order or rank between our clusters. We have actually named them after plants. So our astrocytes are named after herbs, our microglia are named after flowers, and our oligodendroglia are named after trees. And so first I'm going to show you what we see in terms of the changes to abundance of glia in HD. And here is showing you the same UMAP representation of each of our glial types that you've already seen. And then next to it is a bee swarm plot, which shows you the results of our differential abundance analysis. And the populations that are colored in blue are those that are enriched in HD, and those in red are depleted in HD. And what I want you to notice, first of all, is that we see changes across all of the types of glia. We see that microglia, oligodendroglia, and astrocytes are affected in HD. And when we look at the most prominent changes, we can see actually that within each type of glia, there is both an enriched population and a depleted population. And I'm going to go through them each in turn, and I'll start by focusing on the depleted populations, and then I'll come back to the enriched ones. So if we start by looking at the microglia, we can see that there are two very prominently affected populations. There's an enrichment of microglia violet and a depletion of microglia rose. And when we look at the marker genes for these clusters, we can see that microglia rose is identified by some genes that are commonly expressed in homeostatic microglia. And so the fact that these are depleted in HD might suggest that there is a subset of microglia that transition into an activated or phagocytic state in the context of disease. And this has been shown previously that there is a role of microglial activation and inflammation in the context of HD. So we think that's reflected here in the depletion of microglia rose. And then when we look at the oligodendrocytes, or the oligodendroglia rather, we can see that there is an enrichment in the population OPC pine and a depletion of the population oligo-birch. And when we look at the genes that characterize oligo-birch, we noticed two really interesting ones, being opalin and FMN1. And opalin is a gene that has previously been described both by ourselves and other groups as a marker of a specific subset of oligodendrocytes, and this subset has been described as an intermediate oligodendrocyte state, or newly formed oligodendrocyte state, or a myelin-forming oligodendrocyte. And so together with the expression of FMN1, which is a cytoskeletal protein, and rearrangement of the cytoskeleton is a really important part of myelination, we hypothesize that this population oligo-birch represents a population of actively myelinating oligodendrocytes, and this in turn would be consistent with the myelin deficits that are seen in HD both in animal models, in patients, and in pre-manifest individuals. And then moving on to the astrocytes, we see here as well that there is one population that is enriched, astrotime, and a depleted population that we have called astro-oligo. And we've called astro-oligo that because it is a population of astrocytes that also expresses some very typical myelin genes like PLP1 and MBP. And this has been published previously, and it's something that we have seen across different data sets and in experimental models. And we've looked at it at the ultrastructural level, and we've seen that there are some of these cells with an astro-oligo identity that are located near the vasculature, and that they might play a role here. And this has also been shown in the context of HD, that the vasculature is affected, so perhaps that's reflected here in the depletion of astro-oligo. And so I promised you that I would move on to talk about the enriched populations. And although I didn't mention it, you might have noticed as I was going through that the enriched populations are all characterized by expression of molecular chaperones. And we noticed that when we were looking at the markers for these three populations, microglia violet, OPC pine, and astrocyte thyme, that these were characterized by molecular chaperones. And so we looked into this in a bit more detail by looking across all of their marker genes. And we looked for an overlap where we found that there were actually 72 genes that were shared markers between these three clusters. And so we computed that in turn into a protein-protein interaction network, and we looked for functional enrichment of geoterms within that network. And what we found was that the most significant terms were all related in one way or another to protein folding. And when we looked at the term chaperone-mediated protein folding itself, we could see that there were 10 genes that were markers of each of these three clusters, or all of these clusters rather. And they are all genes that are encoding molecular chaperones, or their co-chaperones and cofactors. And molecular chaperones, as some of you might be familiar with, are really key players in the normal functioning of cells. Their role is to contribute to maintaining normal proteostasis, in that they fold newly synthesized and unfolded proteins, or proteins that have been misfolded, and their role is to fold them into their correct conformation or otherwise tag them for degradation by the proteasome. And the third outcome of this, if that fails, is that the protein is allowed to aggregate, and we know that that can lead to cell death, which can manifest as neurodegeneration. And we know, of course, as has been highlighted in many talks during this meeting, that aggregation is a key component of the pathology in HD. So we think perhaps that the upregulation of molecular chaperones selectively in populations that are enriched in HD might represent a survival mechanism, that those cells that are able to upregulate their molecular chaperone machinery to maintain proteostasis in the context of Huntington aggregation are able to survive. And so moving on from this cross-glial signature that we see as a result of our differential abundance analysis to one of the most striking findings from our differential expression analysis, and one that is specific to oligodendrocytes, namely the upregulation of phosphodiesterase 1A, or PDE1A. And PDE1A is a cyclic nucleotide phosphodiesterase, which belongs to a large family, but it's fairly unique in that it is responsive to calmodulin and calcium, and it has a dual affinity for cyclic AMP and cyclic GMP, but it being slightly higher for cyclic GMP. And its role is to hydrolyze these two cyclic nucleotides into their respective monophosphates, and through its regulation of their levels, it can also affect the downstream pathways of cyclic AMP and GNP, which are numerous, and this list is not exhaustive, but we know that they can regulate protein kinases, phosphorylation, the proteasome. They can interact directly with ion channels, as well as acting as second messengers in a number of signaling cascades. And we became interested in PDE1A when we noticed that this was perhaps the most prominent pattern of dysregulation across clusters and across regions within any of our glial types. And as you can see here on this volcano plot, although it is a bit busy, I hope you can appreciate that PDE1A is upregulated in a number of different clusters and across all of the brain regions that we've studied. And when we looked at this in the context of the wider data, including all of the cell types, we do see that PDE1A is expressed in other cell types, especially neurons, but the upregulation that we see is highly specific to oligodendrocytes. And when we look at the individual clusters of oligodendrocytes, we can see across all regions of the brain that PDE1A is upregulated in HD, shown in green, compared to control, shown in blue, in all of the clusters except two, those being oligo-ASH and oligo-Rowan. And in the case of oligo-ASH, we do see that this is upregulated in some regions of the brain, but not all. Whereas in the case of oligo-Rowan, this is actually the only cluster where we don't see a change in PDE1A in HD, and we think that, as you can probably appreciate, that this is at least owing to the fact that there's a higher level of baseline or expression of PDE1A in the healthy brain. And when we group all of our oligodendrocyte clusters together and we just look at the difference between the different brain regions, we can see that PDE1A is upregulated across all of the 4 brain regions that we've studied. And so we find this really robust and consistent pattern of upregulation of PDE1A in Huntington's disease really fascinating, also in the context of what's known about phosphodiesterases previously. And this has led us to hypothesize that this could be something to explore as a future target for therapeutics targeting oligodendroglial functioning in HD. And our rationale for that comes from a few different areas of research. So firstly, we know that phosphodiesterase inhibition generally has a beneficial effect on oligodendroglial functioning. And this has been shown using inhibition of a number of different phosphodiesterases, that this has been shown to promote oligodendrocyte differentiation, myelination, and also remyelination in the context of disease. And then, as some of you might be familiar with, there was a trial of phosphodiesterase 10A inhibitor in HD, which came about due to animal research showing that inhibition of PDE10A could ameliorate some of the striatal pathology in HD mice. But in the trial, this failed to show any therapeutic benefit. But we find it pertinent to note that this was targeted at neurons in the sense that that's where the effect was shown in the animals. And as Anna has mentioned, glia are a much more tractable target for therapeutics because of their ability to self-renew. And then in the case of PDE1, this hasn't been studied widely in the brain or in the context of neurodegeneration, but it has been shown that its inhibition has a beneficial effect in the heart, where in a model of cardiac proteinopathy, this was shown to improve cardiac functioning and survival in a mouse model. And interestingly, it was found that the effect of PDE1A was mediated by its ability to regulate the proteasome. And then finally, just to note that inhibition of phosphodiesterases is something that's being explored as a therapeutic avenue at the moment for neurological disease. For instance, there's an inhibitor of PDE3 called cilostazol that is currently in trial for lacunar stroke. So this is a strategy that's being pursued. And so together with our really robust upregulation of PDE1A in HD, we think that this might be something to explore as a treatment to ameliorate the oligodendroglial pathology. And just very simplistically, the way that this might work is that we see this upregulation of PDE1A in HD, which in turn would reduce the levels of cyclic AMP and cyclic GMP that are available in the cells, and that would impair the function of their downstream effectors, one or more of those, and that in turn this impairs the functioning of the oligodendrocytes. And then of course with treatment we might reverse that with an inhibitor to lower the levels of PDE1A and thereby restoring normal levels of cyclic AMP and cyclic GMP, and also the functioning of their downstream effectors, which might then rescue the oligodendrocytes function. And so just to summarize, we have shown that there is prominent changes to the abundance of all glial types in HD, and we see that some of that, specifically the enriched populations, are characterized by an upregulation of molecular chaperones, and we think that this could be a protective mechanism. And then we also see a really prominent upregulation of PDE1A in oligodendrocytes, and we speculate that that could be something to explore for future therapeutics. And then we haven't really spoken much about this today, but we included 4 different brain regions because we wanted to see whether we could find glial pathology throughout the brain, and we did. And while there are certain subtle regional differences in the glial pathology, we see that this is a global brain disease. And so with that, I'd just like to thank you all for your attention and thank the organizers for inviting us to speak and everyone who has been involved in this project, and to just highlight that this work is also now available as a preprint on bioRxiv. Thank you.
Excellent. Thank you for this excellent presentation. We have time for some questions.
Mostly really a comment. Brilliant work. Thank you. I also want to say it's really great that you chose cerebellum as part of the brain to study, because we have been not really paying too much attention to that part of the brain, which makes me think about using the cerebellum as a reference region in the PET study that was mentioned previously. So just to bring it up as part of the discussion. Thank you.
Yeah,
Thank you.
Hi, thank you very much for a great presentation. Very interesting. One question. You consider the upregulation of heat shock proteins, chaperones, as a protective mechanism. Why wouldn't upregulation of PDE1 not be a protective mechanism and be detrimental?
That's a good question. I think one of the rationales for believing that is, as I showed you, all of the work that's been done with phosphodiesterases in oligodendrocytes generally show that inhibition has a beneficial effect. And so we might expect that having higher levels of phosphodiesterases, including PDE1A, would be detrimental.
But what we will do is overexpress it in oligodendrocytes and find out. So that's ongoing.
Maybe you might have mentioned this, but what's the specificity of PDE1A? Is it only expressed in oligodendrocytes or?
No, so it is expressed in neurons, very highly expressed in cerebellar granule cells, but also in some other neuronal types. And it's found in actually throughout cell types here and there, but very prominent in neurons. And so the specificity is really in the upregulation. It's only in the oligodendrocytes that we see a change.
Great. I think with this, we'll bring the session to an end. Thanks to the speakers, and thank you all for attending.
Mentre tu eserciti il libero arbitrio, scegliendo il tuo preferito tra i vari cibi industriali, e la marca dei tuoi cereali.